Good morning, students. Today we are going to discuss about a very important topic of your syllabus, that is chemiosmotic theory. As we all know that why we take food. We take food to get energy. We take food and that food get digested, and after digestion the food will be absorbed and will transmitted through our blood to different organs, to different cells. So, what will happen inside the cell? Inside the cell, the food will get metabolized, that is catabolized. As we know, the catabolism is a part of metabolism, and after catabolization, from the food particles, some molecules will be released, which finally will get completely oxidized somewhere inside the cell to liberate energy. Do you know which part of the cell is responsible for liberating that energy? That is mitochondria. So, mitochondria, what the mitochondria is? The mitochondria is, we call it as the powerhouse of cell, as it produces energy in the form of ATP. That energy actually is coming from our food. So, the ATP, that is adenosine triphosphate, how it gets synthesized inside the mitochondria. Actually, the whole process of synthesis of ATP occurs inside the mitochondria, and inside the mitochondria there is a system who produces ATP, and that ATP is used by us for different organisms, to do different activities for carry out our day-to-day life. So, now we are going to discuss about how this ATP is synthesized. Actually, the ATP is synthesized the process that is called phosphorylation, and this process also coupled with another process, that is oxidation. That is why the whole process of ATP synthesis which occur inside the mitochondria, we can call it as oxidative phosphorylation, which occurs basically in the inner mitochondrial membrane. As in the previous classes we have studied how mitochondria looks. Actually, the mitochondria is having an outer membrane and an inner membrane. So, inner membrane is having all the machinery for synthesizing the ATP. Now, here we are going to discuss about how this ATP is synthesized by some biochemical process, and this whole process was described by Peter itself as chemiosmotic theory. So, as we know, whatever the food is, whether it is fat, whether it is carbohydrate, whether it is protein, all will get metabolized into acetyl coenzyme that we have already studied. So, that acetyl coenzyme will enter into the acetyl coenzyme will become what we know as oxaloacetic acid inside the citric acid cycle inside the mitochondrial matrix. Inside the mitochondrial matrix, we already have studied that Krebs cycle will occur, and as a result of the Krebs cycle, what will be produced? NADH, FADH, those will be produced. So, those FADH or NADH will finally enter into the inner mitochondrial membrane to carry out go through the electron transport system, and finally will produce water and ATP. So, and this process is very important, because when I will show you what will happen. So, here you can see this is the electron transport system which is having various components. Actually, there are 4 components here 1, 2 is not showing here, I will discuss in the next slide. 1, 3 and 4, actually there is another one which is here. So, 1, 2, 3, 4, these 4 complexes are present in the inner mitochondrial membrane, which will finally produce ATP. Here we can see, which will finally lead to the production of ATP, phosphorylation of ADP to produce ATP. Therefore, this process is called phosphorylation. But now we should know why it is called chemiosmotic theory. Now we are going to discuss about that. Actually, whatever synthesized in the Krebs cycle, that is NADH, FADH2, as I told you, all will come to the inner mitochondrial membrane, and they will donate their electron to the this complex 1. What is complex 1? These complexes are nothing but these are the multi-subunit complexes which are made up of many proteins. Each complex is having many proteins. So, these complexes are very important because each and every complexes are responsible for getting electron and giving electron. That means both oxidation and reduction reaction will occur in each and every complex present in the inner mitochondrial membrane of the electron transport chain present in mitochondria. So, whatever energy will accumulated due to the metabolism of different fruit products, all those energy will come here and those molecules will liberate energy and will donate it to the complex number 1. Complex number 1 will give this electron to ubiquinone, you can see here Q, and those electron will go to complex number 3. As the complex number 1 will donate electron, the complex number 1 get oxidized and complex number 3 get reduced. Then from 3, the electron will move to complex number 4 and complex number 3 will get oxidized and complex number 4 will get reduced. That means it is an redox reaction. Oxidation reduction will occur. And finally, the complex number 3 will donate the electron to, we can see here, we can see the electron to the oxygen, and that oxygen is the final acceptor of the electron which was coming from the molecules produced during metabolism. So, the final electrons will be accepted by oxygen and oxygen become negatively charged and it will combine with the proton to form water molecule. And in each and every step, when the electrons move, some amount of energy will be liberated and that energy will give the inner side of the inner mitochondrial membrane in the matrix. What will happen? The protons. So, here you can see actually this is the matrix and this is the intermembrane space. So, when the electrons move, due to the movement of the electrons, an energy will be liberated and that energy will throw proton through this every complex and proton will come out between the outer mitochondrial membrane and inner mitochondrial membrane. That means in the intermembrane space there will be some gradient of proton. That means a large amount of proton will be there in the intermembrane space, and due to this amount of proton there will be a force that is called proton motive force. You can say proton gradient will be generated here. Why this proton comes here in the intermembrane space? Due to the transfer of electrons from one complex to another. So, that energy from electron transfer will, what will happen? Will force the proton to comes out of the matrix to the intermembrane space. Okay? And this force is called proton motive force. As I told you, I should mention about the complex number. There is another complex that I will tell you. Here is another complex that is called complex number 2. This complex number 2 is not shown here. I will discuss later on, but just I want to mention here. The complex number 2 will get electron from FADH. Here we can see complex number 1 is getting electron from NADH, while the complex number 2 will directly get the electrons from complex number from FADH2. So, similarly, the complex number 2 will get electron from FADH, that will be transferred to complex number 3, then 4. That will also, the transferring of electrons from 2 to 3 to 4 also will generate some energy which will also push the proton out from the matrix to the intermembrane space. So, from all these things, a proton motive force will be produced here. And there will be some potential difference between inside and outside, we can see. So, finally what will happen? There is another complex that we called is an ATP synthase or synthetase. So, that complex is having F0 and F1 particle. This complex is responsible for ATP synthesis. So, what will happen? Finally, these protons will form a gradient and finally through a pore in the F naught particle, the protons will come out with a great force, okay, to inside of the matrix, and that force will help to produce ATP from ADP plus phosphate. So, ADP and phosphate unite to form ATP due to proton motive force that is generated due to sudden influx of proton to inside the matrix through the F naught particle, and due to this force in the F1 particle ATP gets synthesized. Okay. So, we can see that there is an osmotic difference in the H plus ions outside and the inside, therefore, it is chemiosmotic. As here the electrons move from one complex to another, we can say it is an example of redox reaction. As final acceptor of electron is oxygen, we can call it the reaction is oxidative, and also this get oxidized, then it get reduced, when it get oxidized it get reduced. So, that is what? That is, it is an oxidation reduction reaction, as well as it is a phosphorylation reaction due to the production of ATP. So, as a whole we can say it is an oxidative phosphorylation. So, student, can you just imagine what happening here in the electron transport chain? So, this is a brief discussion about the chemiosmotic theory of ATP synthesis. In the next class we will go further and we will discuss more about this. Thank you.